I request Swarnajit Singh, please start. Yes, Swarnajit Singh, please.
Sir, you're not audible.
Okay. So, I will request.
Yes. I'm okay. I'm okay now.
Okay.
Thank you, Dr. Deshmukh. I'm very happy to introduce Dr. Kavleen Kaur, who is the first candidate to present her PhD work on this MBSI platform. I welcome you, Kavleen Kaur. She is assistant professor in microbial technology in the Punjab University. She will present her PhD work to us. We are all very happy in Chandigarh that she is the first candidate from Chandigarh to present her PhD work. Please go ahead.
A very good morning to everyone. First of all, I would like to thank Dr. Swarnajit Singh sir and all the members of Microbial Society of India for giving me this opportunity to present my PhD work on this prestigious platform. I feel so honored to be a part of this webinar series. So I want to thank you all for this opportunity. Should I share my screen now?
Yes.
I request all to mute your mic. Yes.
So my screen is visible?
Yes. Yes, it is visible.
Okay, sir.
Yeah, now it is full screen. Very good. You can make it full screen. Yes.
Okay, so my topic of my PhD work was multicopper oxidase from Acinetobacter baumannii, heterologous expression, characterization, and role in virulence. And I've done my PhD from Department of Microbiology under the supervision of Professor Prince Sharma, Department of Microbiology, and Professor Meena Kaplash, Department of Biotechnology, Punjab University. Coming to introduction of Acinetobacter baumannii, A. baumannii has been recognized as the dreadful gram-negative pathogen responsible for major nosocomial outbreaks and epidemics worldwide. Commonly isolated from the hospital environment, it is the major cause of healthcare-associated infections. However, reports of community-acquired pneumonia, especially in individuals with history of alcohol abuse, smoking, diabetes, and obstructive pulmonary complications are also seen. A. baumannii has been sometimes, it was also known as Acinetobacter because of the prevalence of this pathogen during the Iraq War among the injured soldiers who were passed through several medical facilities. The Infectious Diseases Society of America has grouped A. baumannii among the escape pathogens based on the ability to abscond the action of current microbials. The global spread emergence of multi-drug resistant strains of A. baumannii, scaled as MDR, XDR, and PDR, are of great concern for the scientific community. The WHO has given a priority pathogen list by addressing the issue of global drug resistance, and they have listed this multi-drug resistant pathogens into 3 groups: priority, critical, high, and medium. And out of that, Acinetobacter baumannii, especially the carbapenem-resistant A. baumannii, has been considered to be at the top member of the critical priority group, against which novel drug targets are urgently required. Even COVID-19 has created a perfect storm in the list of multi-drug resistant pathogens. As we know that more than 80% of the patients who were hospitalized with COVID-19, they received an antibiotic therapy, due to which there was 15% increase in the antimicrobial resistant infections and deaths were encountered in the year 2022, in the year 2020, according to the report given by CDC. So what are the factors which have contributed to the resilience of A. baumannii? This is the wide array of virulence factors which are being expressed by this pathogen, its multi-drug resistance, and its high persistence. All of them contribute for making this pathogen a robust pathogen. Then coming to the antivirulence strategies, despite the advances made in the field of genomic and molecular biology for the exploration of the genes that may encode for the virulence factors of A. baumannii, but only functions of only few have been annotated in detail. Multicopper oxidases have been emerged as a recent virulence factor in many pathogens such as Salmonella enterica, Pseudomonas aeruginosa, Cryptococcus neoformans, and Xanthomonas campestris. But its role in Acinetobacter baumannii was not known. Now, what are multicopper oxidases? These are a family of copper-containing enzymes that includes lacases, ceruloplasmin, ferrioxidase, ascorbate oxidase, and nitride reductase. They catalyze the 1-electron oxidation of the variety of substrates along with the 4-electron reduction of oxygen to water. Multicopper oxidases, these are the copper-containing enzymes, and they are composed of three type of copper centers: type 1 copper center, type 2 copper center, and type 3 copper center. The type 2 and type 3 copper center, they together makes a trinuclear cluster. Type 1 copper center is the site where where substrate binds and it is oxidized into its oxidized form. The electrons which are released from the type 1 copper center, they are then transferred to the TNC site where the reduction of oxygen to water take place. What are the possible ways, what are the possible roles of multicopper oxidases in microbial physiology? The first very important role is copper tolerance and hemostasis. Multicopper oxidases have known to have a particular role in the copper hemostasis machinery. It is also involved in manganese oxidation in certain bacterial species. It is also involved in nitrite respiration or denitrification, and also has a wide role in the in stress adaptation and defense mechanism. Now, the functions of multicopper oxidase which has contributed towards microbial pathogenesis, that involves iron uptake and acquisition. Multicopper oxidases play a very important role in the uptake of iron by converting iron 2 into iron 3 forms in various bacteria and fungi. It has also has a role in the formation of pigments, which is the process of melanogenesis. The melanin pigments have been formed in various fungus species, which has contributed it as an important virulence factor. And finally, its copper resistance has a great role, due to which there are certain bacteria like Salmonella enterica and Xanthomonas campestris, which have gained virulence in their microbial pathogenesis. Now, the research gaps was that there was lack of knowledge about the presence of multicopper oxidase in A. baumannii, and its possible function in the physiology and pathogenesis was yet to be explored. So with this, we proposed these 8 objectives for my PhD work, and I'll be explaining them one by one in detail. The first objective was to search NCBI database for the presence of multicopper oxidase in the proteomes of sequenced A. baumannii strains. For this, NCBI database was used, and the completely sequenced strains of A. baumannii was downloaded in FASTA format from this database, and those downloaded proteomes were then further analyzed thoroughly for the presence of multicopper oxidases. Then, since the presence was found to be around more than 98% conservation among the strains of A. baumannii, then multicopper oxidase encoded as COP-A of A. baumannii 80cc19606 was selected for a study, and this multicopper oxidase was designated as ABMCO. The second objective was to predict the conserved domains, 3D structure, subcellular location, and physicochemical characterization of A. baumannii using bioinformatic tools. The results of BLAST-P and conserved domain analysis, they revealed that multicopper oxidase of A. baumannii, it belonged to copper-resistant A superfamily, and it's cupridoxin and contain all the 3 cupridoxin domains, type 1 copper centers and type 2 copper center and type 3 copper center. Further, multiple sequence alignment was used as a tool to check the conservation of this protein with the other strains of A. baumannii. So all those strains which were downloaded from NCBI, they were aligned using multiple sequence alignment. And as you can see in this figure, more than 98% conservation was seen among the strains of Acinetobacter baumannii. Then this is the multiple sequence alignment of ABMCO with other species of Acinetobacter, and we can see 70 to 97% conservation among the different species of Acinetobacter. Then we also aligned the multicopper oxidase of A. baumannii with other bacterial pathogens like Pseudomonas aeruginosa, Mycobacterium tuberculosis, S. aureus, C. jejuni, E. coli, and Salmonella enterica. And though we can see here that the conservation was seen to be moderate, moderate conservation was seen among other bacterial pathogens, but we have a region of this HCH region, histidine, cytosine, and histidine region. This region is conserved among all these bacterial pathogens possessing MCO. So this is the region of the copper binding site, and which is present inside the cupridoxin domain. And this is basically that active region of this enzyme, which was present or it was conserved among all the bacterial pathogens. Then the similarity of ABMC with other bacterial pathogens was also analyzed by 2 BLAST-P tool, and this gave us information that multicopper oxidase of A. baumannii is showing its maximum similarity with the multicopper oxidase of Pseudomonas aeruginosa, which is also an important gram-negative escape pathogen. So it shows its maximum similarity with that of COP-A of Pseudomonas aeruginosa. Further, we predicted the secondary structure of this protein using GOR4 and FIRE2 software. and its tertiary structure was predicted from Swiss model using rigidoporous lignosus lacase as template, and it showed its maximum identity of 28.7% with a sequence coverage of 67%. Then the structure of this ABMCO was validated with Ramachandran tool, and this revealed more than 87.4 residues of the predicted structure were in the favored region. Further, the tertiary structure of this protein was improved using mod refiner server, and it was validated again by Ramachandran plot, which represented 88.6% of the residues in the favored region. Then the quality of this structure was finally checked by ProQ online server, which predicted certain LG scores and MaxSub scores of this model, and they gave us the overall score, gave us an idea that this structure is of good quality and it could be further used for your other bioinformatic analysis. All the physicochemical parameters like the molecular weight, the PI, extension coefficients, half-life, and instability index, they were calculated using PROT-PARAM, XPASI PROT-PARAM tool. And for the subcellular location of ABMCO, a P-SORT-B tool was used, which predicted that this multicopper oxidase has a periplasmic location inside the cell. Then, and a signal peptide, the presence of signal peptide was observed in this multicopper oxidase, and it has a cleavage site between the 23 and 24 amino acid. Further, its secretion, secretory pathway was predicted using SEC pathway as a secretionary pathway for the translocation of the protein. It was observed that the amino acid composition of this peptide, it is not recognized by the secretory machinery of the E. coli, due to which it resulted in the failed translocation of ABMCO to the periplasmic and represented the cytoplasmic localization. Just a minute, please. Further string analysis of ABMC was performed, with which we could, it predicts us with all those interacting proteins which are known as the functional partners of ABMCO. And it was found that most of the proteins were like copper resistant protein B. We have nitrate and nitrite transporters, cobalt, zinc, cadmium, efflux system components, manganese transporters, and acid resistance protein. So most of the proteins which were interacting with the ABMCO, they have their role in either the metal efflux systems, or they were metal transporters and were playing a role in the metal hemostasis. The third objective was to check the prevalence of this protein in the clinical isolates of A. baumannii. For this, the different clinical isolates of A. baumannii were recruited from GMC at Sector 32 and PGI Sector 12 Chandigarh. And the presence of multicopper oxidase was checked using colony PCR, in which an amplified gene of 1.9 kb was observed among all the clinical strains that we recruited from these hospitals, and this gives us an inference that this protein is also conserved among the various clinical isolates. Fourth objective was the heterologous expression of multicopper oxidase of A. baumannii. And for this, we used PET28A as a vector. So we restrictly digested our vector with restriction enzymes BAMH1 and XH1, and we ligated our amplified ABMCO gene into that vector, and then transformed it in our heterologous host, which was E. coli BL21. And the positive transformants, they were confirmed using, again, colony PCR. So this gel picture is giving us the results of this cloning of E. coli in cloning of ABMCO in E. coli. The lane 3 is representing our PET28 vector. The lane 4 is giving the band for the digested vector. Then lane 5 is the vector, which is the recombinant vector, which has the gene of interest. And then once again we digested it with the same set of restriction enzymes, we can see the two bands representing one of a vector and the other of our gene of interest, which we have cloned into it. And seventh one, it is the colony PCR. The band is representing the amplified gene from the positive transformant. Further, the expression of ABMC in E. coli was studied using IPTG. For this, various optimization studies was carried out in which the different concentrations of the IPTG, the temperature, and the time of incubation was varied. And based on those optimization studies, we came to the conclusion that multicopper oxidase of A. baumannii was best expressed, and it gave its maximum protein in the solubilized form using 20 micromolar IPTG at 20 degrees Celsius and for 16-hour incubation. Further, the next objective was the purification of this protein and then characterization of recombinant MCO protein. So this protein was purified in its soluble extracts using nickel NDA affinity chromatography. And then the purified protein, it migrated as a single band of 72 kDa on STS page. Then next was the characterization of this recombinant protein. So first in activity, since this is an enzyme, so we the first aim was to perform its activity assay. For this, the oxidase activity of purified ABMCO was determined in 100 millimolar phosphate buffer using glycol as a substrate, but no activity was detected at all. Then we exogenously added some amount of copper sulfate into our assay mixture, and we got these results that ABMC could be expressed, or it showed its activity even in a trace, even if a trace amount of copper, for example 1 micromolar copper was added, and the activity was seen. And the maximum activity, it was observed at 50 micromolar concentration of copper sulfate. So all those multicopper oxidases whose oxidative activities are copper dependent, they have been categorized as pseudolacases. And all the bacterial tolerance proteins which have been studied till now, for example, that from Pseudomonas syringes and Pseudomonas campestris, E. coli, Mycobacterium tuberculosis, Campylobacter jejuni, and Brucella melanoensis, all of them fall under the category of pseudolacases. All of them show their expression only when they are being expressed in the presence of copper ions. Then the effect of pH on activity and stability of the ABMCO was studied, and it showed that the maximum activity of this protein was seen at pH 6, and 80% activity was at pH 7. However, then decrease in activity were observed in both the acidic and basic ranges. ABMC was completely stable at pH 6 for 1 hour and retained 80% activity up to 2 hours and 65% up to 5 hours in the pH range of 6 to 8. After 24 hours of incubation, it retained around 15% of the activity in the range pH 6 to 8. So this gives us a conclusion that this protein of multicopper oxidase has its stability in the pH range of 6 to 8. Then the effect of temperature on activity and stability was checked. The maximum activity was observed at 50 degrees Celsius, and it showed 30% activity at 37 degrees Celsius. And thermostability studies revealed that this protein retained 40% activity up to 4 hours in the temperature range of 37 to 45 degrees Celsius. And it retained only 18% activity after 1 hour of incubation at its optimum temperature. So it showed a good activity in the temperature range of 37 to 45 degrees Celsius. Then the effect of certain metal ions were checked on the activity of ABMCO, and all the metal chlorides or metal sulfate we checked, they led to the decrease in the activity of ABMCO, except for NaCl and KCl. It was found that in the presence of NaCl, the activity of ABMC was increasing gradually, and the maximum was seen at 1 molar concentration. And even at 1.5 molar concentration, the activity was slightly reduced. And the same case was seen with KCl. 3 times increase in the activity of ABMCO was observed at 1 molar concentration, and by further increasing the activity towards the concentration towards 1.5 molar and 2 molar, even a slight reduction in the activity was seen. Then the effect of certain inhibitors were checked. L-cysteine at a concentration of 150 micromolar completely inhibited the activity of ABMCO. And sodium fluoride and sodium azide, they inhibited the activity at a higher concentration. So this gives us an idea that usually the other multicopper oxidases, which are of industrial importance or of other bacterial pathogens, they are inhibited at a very low concentration of sodium fluoride and sodium azide. But multicopper oxidase A. baumannii, it even tolerated the very high concentration of these inhibitors, and the high concentration was required to inhibit the activity. But with L-cysteine, its activity was inhibited completely at 150 micromolar. So it was proved to be a potent natural inhibitor of ABMCO. Then the kinetic properties of ABMC was studied using Michael-Menten equation, which showed its maximum catalytic efficiency of this protein towards ABTS as the substrate. Next objective was effect of medium components and other stress conditions on the production of MCO. So first of all, we subjected our culture with nutritional stress. We modified the media of the minimal media which we were using for the production of this protein M162. So we modified, we provided the stress in terms of the medium component stress. For example, firstly, we tried with phosphate stress, and it showed that A. baumannii, it could tolerate a high level of phosphate stress, as the complete inhibition in the production was seen at 250 millimolar phosphate buffer. Then for nitrogen stress, we checked its activity, its production in the presence of yeast extract. And it was shown that 0.3 to 0.4% of the yeast extract, it showed its maximum production, but the inhibitory concentration was again a much higher, that is 1.5% of yeast extract. And similar results were seen with tryptophan as a source of nitrogen, that it could tolerated around 1.2% of the tryptophan in the media, and its inhibition was seen at 1.5% tryptophan. Then for the carbon stress, the multicopper oxidase was produced in M162 media by supplementing a glucose concentration. It was seen that by adding even a 0.1% of glucose, there was around twofold increase in the production of ABMCO. But at 0.2%, its production was completely inhibited, which could be due to the phenomenon of catabolite repression. Then effect of other stress conditions on the production of ABMC was also checked. For this, we took idiophysic phase of this culture, and then we subjected it to different stress conditions for 48 hours. And after that, the activity of multicopper oxidase was checked in the presence of copper ions. So firstly, we subjected our culture to osmotic stress, and for this we chose NaCl, sorbitol, and polyethylene glycol as the sources of osmotic stress. In the presence of NaCl, it was found that the activity, the production of ABMC was increased up to the maximum, that was 2.4-fold increase was seen at 700 millimolar concentration of NaCl. And thereafter, the decrease in the production was noticed. But still, we can see that even at a high concentration of 1 molar, it showed twofold increase in the production of this protein. With polyethylene glycol, only 20% increase in the production was observed at 5%. And with sorbitol, the 20% increase was seen at 750 millimolar concentration. So this bacteria, the production of this gene was increased in the presence of NaCl stress.
Are you talking the recombinant enzyme or native one?
No, sir, this is the native one. We are modifying the cultural conditions of A. baumannii.
Exactly. But previous slide was related to the recombinant one.
So we have characterized all those activities which we have performed. They were for the recombinant protein, but the stress
Exactly.
conditions were given to the A. baumannii, the
Yeah, that is
culture
what.
only.
Please go ahead.
Yes, sir. So for oxidative stress, the culture was subjected to different concentration of hydrogen peroxide or menadione, but no change in the production of MCO of A. baumannii was observed under oxidative stress conditions. Then we subjected our culture to different metal stress. And for this, first of all, copper ions were used. And we can see that increasing the concentration of copper sulfate as a stress factor in the culture media led to the gradual increase in the production of ABMCO. The maximum increase, that was 3.5 fold, it was observed at 1500 micromolar concentration. And even at 2000 micromolar concentration, the activity was threefold higher than the control or the unstressed cells. So this gives us that the production of ABMCO is increased tremendously in the presence of copper stress conditions. Whereas under iron stress, the decrease in the activity was, increase in the production was seen. Similarly, when we exposed our culture to heavy metal stress like cadmium ion, again, this resulted in the decrease in the production of ABMCO. But the presence of manganese ions, they did not affect the activity significantly. Further, we also subjected our culture to different antibiotic stress. For this, the culture of A. baumannii was treated with sub-MIC concentrations of the different classes of antibiotics. And we chose two antibiotics from each class of aminoglycosides, fluoroquinolones, polymyx, and carbapenem, cephalosporins. But again, we couldn't find any significant change in the production, or there was a decrease in the production of ABMCO. So ABMCO doesn't act as a specific protectant against antibiotic stress. So on the basis of this stress conditions, we got a result that the production of ABMC was significantly higher when we subjected to copper sulfate stress and NaCl stress. So afterwards, we decided to check its expression on the multicopper oxidase gene. So for this, the idiophilic phase of culture of A. baumannii, it was subjected to copper sulfate stress or sodium chloride stress at their concentrations of 1.5 millimolar and 700 millimolar respectively for 90 minutes. And then the total RNA was isolated from both the stress as well as non-stress samples. The RNA was converted into cDNA, and further it was used, the expression was checked using qRT-PCR, using 16S ribosomal RNA as the endogenous control. So results of qRT-PCR, they revealed that a significant upregulation, that is 8-fold increase in the expression of ABMCO, was seen both in the presence of copper sulfate and sodium chloride stress. As we can see in this graph, that the relative expression of ABMCO was 8-fold as compared to the non-stress cell. So this has suggested the protective role of ABMCO under these stress conditions. So the inference that we got from this objective, this is, we know that multicopper oxidases, they are an important class of regulatory proteins and have been playing a role in copper tolerance and copper hemostasis in many pathogens like E. coli, Salmonella enterica, Klebsiella pneumoniae, Mycobacterium tuberculosis, Campylobacter, and Brucella by oxidizing the toxic copper ions to the less toxic copper ions. So qRT-PCR studies of our qRT-PCR studies, they reveal that ABMCO, it is also a copper regulated multicopper oxidases whose expression increases significantly under the conditions of copper stress. So in accordance to these results, we confirm that the multicopper oxidase of A. baumannii, it form an important component of a copper hemostatic machinery, and it explains its survival strategy inside the host and its successful dissemination of this pathogen inside the host by oxidizing the toxic copper ions towards the less toxic copper 2 ions. Apart from this, the production of ABMCO was also significantly higher under the condition of high NaCl concentrations. And for this, ascorbate oxidase is considered to be a major stress response multicopper oxidase against high salinity stress and drought in plants. But among bacterial pathogens, the role of multicopper oxidase in protection of A. baumannii from saline stress, it is being reported till date only in this study. Next objective was the role of ABMCO in the survival and virulence of A. baumannii in terms of metal hemostasis. For this, we checked its copper resistance and hemostasis. We checked its property for iron uptake and acquisition that was performed using ferrooxidase assay. Its manganese oxidase activity was performed. The siderophore oxidase activity was performed. And finally, its survival in the macrophages was checked. So for copper resistance, the culture of A. baumannii, it was grown in the presence of increasing concentration of copper sulfate, both in an enriched media and in a minimal media for 24 hours. And in LB, in the enriched media, it was found that the growth patterns was, it was able to grow in the enriched media containing around 1.5 millimolar copper sulfate at a concentration 1.5 millimolar copper sulfate. And then even at 2 millimolar, we can see that its growth is only 20% decrease. So it showed a growth resistant, copper resistant patterns of the growth in LB media. Similarly, in M162 media, it was seen that till 0.5 molar of the concentration, no significant change in the growth was observed. But by further increasing the concentration, it led to the gradual decrease in the growth of this A. baumannii culture, and the decrease was seen till 1.5 millimolar concentration, and the growth was totally inhibited at 1.75 millimolar. So we can see that it is showing good growth in the copper sulfate up to the concentrations of 0.5 millimolar copper sulfate. Then we wanted to check how this the pattern of copper resistant was checked by measuring its growth at different time intervals. And we can see that equal growth patterns was observed at 6-hour and 24-hour incubation from concentration 0 to 1.25 millimolar. No change in the growth patterns was seen. They were showing equal growth patterns. Then A. baumannii, it could not grow in 1.5 millimolar copper sulfate up to 6-hour incubation. As we can see that at 6-hour, its growth was drastically reduced. But a delayed growth was seen in the same concentration at 24-hour. So this has suggested the time-dependent adaptability of A. baumannii to high concentrations of copper. So this gives us to a conclusion that ABMCO is a copper-related protein whose expression confers protection against copper stress, and it is a determinant of copper resistance in A. baumannii. Then it's the property for iron uptake and acquisition that was measured using ferrooxidase activity. For this, the assay was performed using 200 micromolar ferrous ammonium sulfate as a substrate, and it was monitored for the oxidation of Fe2 to Fe3 at 315 nanometer, but no increase in absorbance was observed. So this has suggested that ABMCO lacked ferrooxidase activity and it could not catalyze the oxidation of Fe2 ions to the Fe3 ions. Similarly, for manganese activity, the manganese activity was also found to be absent in A. baumannii. Then for siderophore oxidase activity, the purified ABMCO that was assayed using various siderophore analogs, for example, 2,3-DHB as a substrate, and its oxidation was monitored at 400 nanometer. An increase in ABMCO activity was seen in the presence of siderophore. So this confirmed that multicopper oxidase of A. baumannii also possess siderophore oxidase activity. Now we know that siderophores are the iron chelating compounds which are secreted by microorganisms to serve to transport the iron across the cellular membranes. But it has been seen that under the conditions of copper stress, these siderophores, which are the iron chelating compounds, they also act as copper 2 reductants. That is, they will catalyze the reduction of less toxic copper ions to the more toxic copper ions, which may in turn lead to certain cellular damage by generating free radicals such as by conducting the Haber-Weiss reactions and the Fenton's reaction chemistry. Moreover, the siderophore bound copper ion, this complex is manifold more toxic in comparison to the copper 1 ions alone. So the property of multicopper oxidases to oxidize siderophore analogs and or their precursor, this is an alternative mechanism for granting its protection against copper toxicity. So if it is oxidizing the siderophores, that means it is reducing or it is inhibiting the conversion of the copper 2 ions back to the copper 1 ions, which are the toxic one and which could lead to the oxidative stress inside the cell. So this property of MCO of A. baumannii to oxidize siderophore analogs is giving it again its survival strategy to survive inside a host. Also, we have seen that under the conditions of copper stress, when copper it is in high amount inside a cell, it hinders the uptake of other important ions like iron ions or zinc ions. So oxidation of siderophore, it is also an indirect way of maintaining metal homeostasis inside the cell. Because when the siderophores will be oxidized by ABMCO, they will release iron ions. So the release of iron ions, it will maintain the transport of metal ions across the membranes. So this indirectly also it is playing role in a metal homeostasis. So in this regard, we concluded that ABMCO has been able to oxidize 2,3-DHB, which suggests its siderophore oxidase activity, and it could be the survival strategy of this pathogen at the interface of copper and iron ions to maintain homeostasis inside the cell. Lastly, we checked the survival strategy of A. baumannii inside the macrophages. So for this intramacrophage survival assay was performed using raw 264.7 cell lines. These cell lines were infected with ABMCO expressing cells and ABMCO non-expressing cells of A. baumannii at multiplicity of infection 100:1 for 45 minutes. Then the sample were lysed at 0 hour, 2 hour, and 4 hour by aspirating the medium and lysing the macrophages with Triton X in phosphate buffer, followed by the plating on M162 media agar plates for the enumeration of the bacteria that survived. So the intramacrophage survival assay, they give us this result that 5% and 3.4% increase in the survival rate was observed at 2 hour and 4 hour respectively post phagocytosis in case of ABMCO expressing cells relative to the ABMCO non-expressing cells. So we have seen that the cells in which the multicopper oxidase was expressed, they were showing 5% increase in the survival rate at 2 hour post phagocytosis, and only 3.4% increase was seen at 4 hour post phagocytosis. Now the inference that we get from this result, we know that macrophages are the first immune cells that has to be recruited at the site of infection, and they mount a robust antimicrobial response in terms of phagocytosis. In case of A. baumannii infection, this has been proved that the macrophages, they form an important component of early host response. So our results, they give us an idea that A. baumannii, the A. baumannii which were expressing multicopper oxidase, they led to they help the early survival of this pathogen inside the macrophages, and afterwards, after the 4 hours, the role of neutrophils come into action for the subsequent clearance of the pathogens. And it has also been proved that whenever a macrophages it engulf any invading pathogen, then the expression of copper importers like CTR and R1 and copper carrier proteins, these genes are upregulated in response to the invading pathogen, so that the concentration of the copper can increase in the macrophage and they can carry out their oxidative stress and other respiratory burst mechanism. And even the enzymes which are involved for these phenomena, all of them are copper dependent. So the concentration of the copper increases in macrophages in response to an infection. So to counteract the successful pathogens, they are opting for copper resistant strategies so that they could survive inside the macrophage, or they could bypass the phenomena of phagocytosis, and they could disseminate to the other tissues or other organs. So the copper resistance patterns of A. baumannii has contributed to its survival strategy inside the macrophages, but only for the earlier survival, only up to 2 hours. And since no remarkable difference in the survival, in the percentage survival of the cells was seen in comparison to the non-expressing cells, we concluded that multicopper oxidase of A. baumannii, it is responsible for the copper resistance of this pathogen, but it is not required for the virulence of this pathogen. And the last objective was the screening of PubChem library for the potential inhibitors of multicopper oxidase of A. baumannii. So for this, different structures of the natural compounds, they were retrieved from a PubChem library in NCBI. Their active site residues were predicted using 3D ligand site online server, and then further patched-off server was used to find the possible interaction of this protein with the different ligands. So for this, a lot of in silico studies were done, in which we have docked our ABMCO with different inhibitors, with different natural compounds, the substrates, and even the antibiotics. And the results which we got, they give us a conclusion that we know that structural studies, they have been playing a very important role in the field of drug development. And among the various substrates, it showed its maximum score with that of ABTS. The interaction was maximum with ABTS and kojic acid as the inhibitor. And by comparing the results of inhibitors and substrates with the natural compounds, we saw that there are certain plant extracts, plant compounds like silibinin, palmitic acid, tocotrienol, curcumin, thymol, eucalyptal, oleic acid. All of them, they were showing good binding scores of this protein with these ligands. So they were suggesting a strong affinity of these natural compounds with ABMCO. So such compounds may act by competing with the binding sites of ABMCO for their substrate, and could be further used as a drug targets for the inhibition of the action. Similarly, when we compared the result of docking studies of ABMCO with different antibiotic classes, it was shown that the scores of antibiotics were also in the same range as that of substrates. So that was saying that give us an idea that antibiotics are also interacting in a good affinity with that of ABMCO. And the maximum score was seen with carbapenems and colistin. So we could hypothesize that the good binding score of ABMCO with these different antibiotics, it could be related to the emergence of antibiotic resistance in A. baumannii. Like A. baumannii could make some conformational trans changes or transformations in the structures of antibiotic, which could render them ineffective. But further wet lab studies were needed to carry out or validate these in silico studies and to target multicopper oxidase as a drug target for the novel drug therapies. So with this, we concluded our PhD work, and with an aim to explore and the prevalence and role of multicopper oxidase in the survival, its metal hemostasis and virulence. So we concluded that multicopper oxidase was found to be prevalent in the clinical isolate. It was highly conserved among the strains of A. baumannii. This protein exhibited copper resistance and siderophore oxidase activity, which depicted its role in metal hemostasis and survival under metal-induced stress, which are generated by the toxic redox state of iron and copper ions. It lacked manganese and ferro-oxidase activity, which suggested that it has no role in iron uptake and manganese oxidation. Expression of ABMCO was increased significantly under the stress conditions of high copper ions and high NaCl conditions, which suggested its protective role in adaptation and management against these stress conditions. However, other stress conditions like oxidation, antibiotic, other metal ions, they do not affect the production. So ABMCO, it helped in the early survival of A. baumannii inside the macrophages and was not required for the virulence of this pathogen. So we could conclude that ABMCO has a role in the survival of this pathogen inside the host and in the metal hemostasis mechanism. And further, this could be recruited as a novel drug target in future to control its control the survival of this multi-drug resistant A. baumannii in the host. These were my publications, and that was all from my side for this talk. Thank you.
That was an excellent talk, Kavleen.
Thank you, sir.
Excellent
Yes,
talk.
yes. Sorry, sir, to interrupt. Just I received her CV. Hello. Hello.
Yes, sir, you're audible.
Okay. So first of all, I am really thankful to Calvin Kaur for giving a detailed lecture on her PhD topic. And definitely the many research workers have joined today. There will be discussion. Some queries are there from Dr. Prakash Halami. But before that, I would like to tell the academic achievement of Calvin. The best academic achievement, I consider that she is a student of Dr. Pri Sharma. I know his contribution. He is one of the eminent scientists. So really, her work is good, and credit maximum goes to her guy.
Yes,
Then
sir.
she is an expert in molecular biology, basic microbiological techniques, and in bioinformatics. Now she is a very fresh PhD candidate, means she has completed her PhD in 2019. And she is working as a guest faculty in Department of Microbiology and Biotechnology in Punjab University, Chandigarh. Then she has worked on projects at MSc, particularly on Escherichia coli. Then on PhD, she has a good publications. About 7 publications already she had, and in a very reputed journal like Journal of General and Applied Microbiology, and other important journals are there. Second, she has achieved, clarified NET, GATE, and ICMR SRF, Senior Research Fellow she got. She has attended many conferences and presented posters. Now, really, Kelvin, your contribution, your academic achievement is a very good.
Thank you, sir.
So I will tell you, now you come for any one conference for oral presentation. I will be happy to listen you in a conference as an oral presenter.
Thank you, sir.
And I wish all the best to you for your future. Now the topic is open for discussion.
And sir,
So-
I also want to, I feel honored to be a student of Professor Prince Sharma and Professor Neena Kaplash. And I feel so blessed that I've been the student of such privileged scientist of Punjab University. And I feel so honored and thankful to my guides.
Actually, I should thank to Swarnajit Singh sir, because he has given me 4-5 numbers of such best candidates from Chandigarh. Really thankful to Swarnajit Singh sir.
Thank
Yes,
you,
sir.
sir. Thank you.
I also want to thank Swarnajit sir for giving me this opportunity so that I could speak for my project work, for my PhD work on such a privileged platform of India. So it's
Correct.
a great honor for me. So thank
Yeah,
you.
you will get a memento at your home address. You will get a certificate, and this lecture is recorded. So you will get the link, YouTube link of this lecture to circulate among other researchers.
Deshmukh sir,
Now-
Deshmukh
Yes,
sir, I will personally present the memento to her.
very good. Thank you. Thank you. Thank you. Now I will come to Dr. Prakash sir from CFTRI Mysore. And again, I am thankful to him because regularly he is attending lectures of a fresh PhD candidate and encouraging them, inspiring them. I request actually all senior scientists to encourage the young.
Maybe next Monday I will not be able to attend. I think next Sunday, I mean, because I will be in Nagpur for this Agrovision exhibition. Agrovision exhibition is in Nagpur from 25 to 28. I would also join with Dr. Deshmukh to congratulate Dr. Kevin Kaur for nice work. And as Dr. Deshmukh also told, in fact, Dr. Prince Sharma is my very good friend. We worked together in AMI and I visited once or twice Punjab University. And more importantly, candidate has done the very extensive study, 8 objectives. It's a very elaborate work, almost like a 2 PhD thesis, I would say. And she has published more than 5 research papers in a impact factor journals.
Correct.
My 1 or 2 questions were there, which are actually there in the chat box. So if she can read and answer on her own,
Kalvin, whether you can
which are not
read,
very serious questions.
Yes,
Just I
sir.
have put there.
please read the questions in chat box.
Okay, sir.
Can I ask one question to Kavlin?
Yes, sir.
Kavlin, why did you choose this baumannii species?
Sorry, sir, I didn't get it.
Why did you choose the baumannii species only?
Sir, because this is the red alert pathogen which have been given by WHO, the most emerging multi-drug resistant pathogen against we need to develop good novel targets or vaccine targets. So as sir were also working in the field of baumannii, sir has done a lot of good achievements in the field of reverse vaccinology also. So we tried to work on what other virulence factors could be chosen from this pathogen so that we could exploit them for further drug targets or even the vaccine targets. And this pathogen needs, it's among the top list of the WHO against which we require need to study its physiology in pathogenesis in detail, and we need to figure out the better novel drug targets.
Yes, that's very good. Any more questions by anybody?
Yeah, Prakash Alami has written question. I will read it. Relevance of virulence by baumannii and multicopper oxidase is not very clear. He's putting question.
Yeah, in fact, she has explained in a subsequent slides that there is no relevance with reference to the multicopper oxidase. But if she can conclude something, it will be interesting because your title says yes.
We couldn't correlate it better on the virulence strategy, but our studies, they gave us a clue that this is surviving, it's giving its early survival inside the macrophages before the neutrophils come and they come into the action. And further, I feel like further in vivo studies would be a good method for comparing this expression and how it is playing role in the virulence of this pathogen. We performed all the in vitro studies,
and next question was what Dr. Swanjit Singh asked. I just wanted to know whether you checked among the virulent and non-virulent strains of baumannii multicopper oxidase activity.
yes, sir.
So that also actually,
yes, sir.
of course, is not needed, but then if
we have checked it. We, like I told, I have explained in the slides that we recruited some of the clinical isolates of A. baumannii, and all of them were multi-drug resistant. We had already checked them, and all the multi-drug resistant clinical isolates, they were expressing the gene for multicopper oxidases, and we even checked the activities in vitro using different substrates, and they were showing activity. It was like conserved among all the strains that we procured from the drug resistant strains from PGI and GMCH32.
Yeah, as far as the thesis organization, I don't know whether this heterologous expression which you have shown in the objective 6 should have been a little bit later, perhaps. That is what I felt, because you go in the objective 7 with the native organisms, right? Native enzyme.
That was the stress conditions.
So yes. So that should have been the before heterologous expression should have been the subsequent, and then you do the bioinformatics pertaining to identifying the target required, which inhibit the multicopper oxidase.
Yes.
That should have been. That is what actually my opinion.
Yeah? Yes, sir. I'll work on that.
Very good. Are you continuing similar work or anything now? Actually,
no,
Dr. Samir has carried out attended our training program, so I know him. I hope he is a faculty there, right, Dr. Samir?
Dr. Samir,
in the microbial biotechnology department.
He has shifted to Banaras Hindu University now.
Okay.
Okay.
So if you find, please convey my regards to Dr. Prince Sharma.
Yes, sir. Sure, sir.
Dr. Kevlin, yeah. Thank you. Thank you very much.
If there is no other question, then I request Pradeep Kaur to tell the vote of thanks.
Okay, sir. Like I told, it's like been the first and the major opportunity which I got through Dr. Swaranjit sir to present my work on such a good platform, such a privileged platform where I could learn a lot from all those.
Dr. Kevlin, there is one more question actually in the chat box.
Okay, sir, I'll check.
That is from Kavita. Just have a look. Can you please correlate the role of MCO and RND transporter an important efflux pump in A. baumannii? Just have a look to that question.
Yes, sir, yes, I can see that.
Yeah.
Okay, so multicopper oxidase, basically we have the regulator which is for the copper homeostasis in A. baumannii. It encodes for two proteins. One is COP-A, which is a ATPA dependent this efflux protein, and this multicopper oxidase. So what happens actually, like wherever the concentration in the copper, the concentration of copper increases in the cell, what happens? The efflux protein, that COP-A, which is the efflux protein, that will take up the copper ions from the cytoplasm to the periplasm. And in the periplasm, we have our multicopper oxidase, which will be converting those toxic copper ions into a non-toxic copper 2 ions. So basically, all these proteins, they are working in conjunction for the process of copper homeostasis. And I also want to add, like, if we want to study the virulence of this organism, of virulence in terms of these proteins like multicopper oxidase, that it will be more convenient if we study the whole copper regulon. Like, it has been proved that certain efflux proteins like COP-A, they have a role in virulence. So we could just generate certain mutants जिसमें कि हम अपनी multicopper oxidase को भी नॉकआउट करें और साथ में हम उन प्रोटीन्स को नॉकआउट करें, efflux proteins को knockout करे, so that the complete copper agalon is removed, and then we could further take it over to the in vivo studies, now how the virulence is being affected. So they are working in conjunction with these proteins.
Okay, thank you, Kavleen. And now I request Dr. Pradeep Kaur to say the vote of thanks.
Yes, thank you, sir. Dr. Kavleen Kaur, first of all, I would like to congratulate you for working on this very pressing issue of the antibiotic resistance by Acinetobacter baumannii. Dr. Kavleen, I am very eager to know about the status of the phage therapy in India in combating the problem of elimination of this dreadful bacteria. I believe that this bacteria is causing a problem not in the Indian hospitals, but in foreign countries as well. So though it is not directly related to your PhD work, what is the status and how far we have achieved the success in covering this infection caused by Acinetobacter?
Phage therapy has been a successful strategy for combating antibiotic resistance in many pathogens and even in our department, our professor, Dr. Sanjay Chibbar, he's been working on this phage therapy only, and they have given a lot of successful researches to combat again a multi-drug resistant escape pathogen, Staphylococcus aureus. But for A. baumannii, I will do check for it. I have no idea about how far it is successful in case of A. baumannii.
Okay.
So, but I will check on it.
Yes, sure. I would like to listen and have an update on this thing.
Yes, sure. I'll update you.
Pradeep, I request you to say the vote of thanks now.
Yes, sure, sir. Sure. Okay, Dr. Kavleen Kaur, I would like to thank you for presenting your PhD work in just so lucid and an extensive manner, and making us aware that how this multicopper oxidase is playing a very vital role in the infections caused by this bacteria, in the virulence, in the metal homeostasis, and the survival of this particular bacteria. Next, I would like to extend my heartfelt thanks to Dr. Arvind Deshmukh, Honorable President, Microbiology Society of India, for always inspiring us and motivating us and providing us platforms to come together and share our ideas, opinions, and the things that we are all doing on such a big platform and sharing it with all the scientific community around. Next, I would like to thank Dr. Swaranjit Singh, President MBSI Chandigarh unit, for his untiring efforts in order to arrange this webinar series of the PhD scholars. He actually was looking out for some fresh PhD candidates from the Chandigarh region, and luckily been successful in getting Dr. Kavleen Kaur from Chandigarh itself. So you are the maiden speaker from the Tri-City. So it was an honor to have you, to listen to you, to your work. So Dr. Swaranjit sir, he's really put up a lot of effort in arranging this webinar series, and we would just like to and hope to look forward to many more such interactive and informative sessions in the future. Next, I would like to, lastly but not the least, all the esteemed delegates who spared the time to have their presence on board, and that's all from my side. So I'm just very extending my heartful thanks to one and all. Thank you so much.
Thank you very much, Pradeep, for the nice vote of thanks.
Thank you. Thank you all.
Thank you. Thank you.